It's been a real pleasure coming here, and I really thank you and thank everyone who was involved in organizing. And I appreciate everyone staying till the end. I'm going to punish you by talking about two different things today at the very end of the day and trying to ruin your dinner. Okay, so the first thing I'm going to talk about is twins. There we go. So identical twins are they're cute. It's kind of nice that they dress alike and they get married together. They even marry identical twins, which is pretty. Some of them are separated at birth, and you know, we're struck by the similarities between twins. You know, there are good twins and there are evil twins, and but I think what's much more interesting are cases where there are discrepancies between these genetically identical individuals, where there are good twins and bad twins. For example, here, this guy, okay. One of those guys, it doesn't really matter because they're identical. It doesn't make any difference. Am I not pushing the right button?
Twins, right?
Yeah, exactly. Oh, is that?
It's the on button.
What? You need an on button? So this guy these were separated at birth, twins, actually. This guy was raised in Germany and was a member of the Nazi Party. This guy was raised in Trinidad and was raised Jewish, actually. This twin actually is in prison in California for trying to murder that twin. Or maybe it's the other way around. One of them tried to murder the other one. And these guys are twins who are completely different. Well, actually, they're both Danish brewers, but they don't talk to each other, and they have competing brewing concerns. This one is called Evil Twin. So, you know, you can see, they already know who's good and evil in that pair. And what we're looking at are examples of phenotypic heterogeneity, genetically identical individuals who nevertheless are different. Now, where is phenotypic heterogeneity important in TB? We've already heard about it. TB treatment takes a very long time. It takes 6 months. I'm highlighting here the drug rifampin because that's going to come up again. And why does it take so long? We think of most infections as being treated rather quickly, but we already heard from Cliff that there's different degrees of success with different times of treatment, and that there are relapse rates which drop over time. Cliff told us that most people are cured after 4 months, which is true, although those numbers don't really match up with his numbers. Maybe because I made them up. But it requires a full 6 months to really decrease the relapse rate in the entire population. So there's variability. So what is that variability? When you think that's going on? Well, of course, we know about genetic variability, and the classic example of that is HIV, which mutates at an incredible rate so that evolution occurs very quickly and you get antiviral resistance in single viral bursts. TB mutates very, very slowly, though, and while resistance arises, it arises very infrequently, and we don't think that is largely the result of failure in most drug-sensitive TB cases. In fact, when they fail, they usually fail with drug-sensitive organisms. We heard already about penetration into lesions. There are all kinds of different lesions in TB, and the beautiful work that Cliff collaborated with Véronique Dartois on looked at the pharmacology of penetration into lesions, and we know that different drugs get into different lesions differently. In addition, the bugs are different in those lesions. They're different environments which induce different responses in the bugs. So that's playing a part. But I'm going to introduce another idea, which is phenotypic variability in the population. So what do I mean by that? So genotypic variation, getting back to the mutation idea, is in a population of cells. If you have a resistant organism and you treat with antibiotics, you kill everything but the resistant organism. And if you stop treating, or continue treating in this case, what grows back is a population that's completely resistant. However, with phenotypic variation, there may be a phenotypically resistant organism in the population. When you treat, you kill all the other organisms. When you take the drug away, what grows back, though, is the same population as before with a variety of phenotypic variants. Now, the problem is they are genetically identical. So how can you tell genetically identical individuals apart? And that gets to the work of Hesper Rigo. Hesper is a former postdoc in my lab. She's now at Yale, a job she got despite not publishing anything in my lab, which is a testament to her. She's really, really good. Although finally, her paper is, we heard on Friday, is finally coming out. So finally, she's paid her price for being in the lab. She's extraordinary, and she is trained as a microscopist, a physicist at UCSF, actually, and really brought a different dimension into the lab. But before I introduce you to her, I'm going to reintroduce you to something else. You've seen this slide a million times, the distribution of TB in the world. What people don't think about so much is the hidden epidemic, where we have literally ones of cases across the world of Mycobacterium smegmatis infections. Smeg is our sort of workhorse organism, and many people in the audience have heard this before. You tend to think of it as a safe organism, but if you grow up like 10 liters of it and you stick your head in, you can drown. So Hesper asked, what functions vary at the level of an individual cell? And she's looked at lots of things. I'll show you one brief snapshot of them later, but today I'm going to concentrate on one of them, which is a molecule called calcine AM. Calcine AM is a non-fluorescent molecule which gets into cells somehow. It can also get out of cells. And when it gets out of cells, it's still not fluorescent. But if in the cell it encounters a cellular esterase, it's cleaved and becomes fluorescent and is trapped, pretty much trapped entirely inside the cell. So the cells light up once that occurs. So this one molecule allows us to assay a number of different processes at once, uptake, efflux, and the concentration availability of esterases. And so when you stain Mycobacterium smegmatis with this dye, what you see is a huge variety of organisms that have different fluorescence patterns. It's tough to see in this picture, but if you use FACS, looking at the stained cells, the red peak here, you can see that there's a considerable spread in the degree of fluorescence, about 2 logs of spread, in fact, between the brightest and the dimmest cells. And calcine accumulation changes with time in individual cells. So, thank you. It doesn't really look like anything, does it? Okay, let's try this. In one of these movies, using similar systems to what we've seen before, if you watch individual cells, when they divide, the progeny, the 2 daughter cells, have different degrees of brightness. So it changes very rapidly, at least at the time of generation, although it's relatively constant once a cell division has occurred. And we'll come back to that a little bit later. Wait, stop doing that. Okay. Now, that would be kind of interesting, but not so interesting if it weren't for the fact that Hesper found that there was a correlation between this phenotype and drug susceptibility of individual cells. The way she showed that is to look at rifampin susceptibility of these cells. What she did is she labeled the cells with a fluor, and then added rifampin, and then followed individual cells to see whether they divided after rifampin therapy. And what she found was that the very brightest cells, which are the ones over here, are failed to divide. So these guys were being killed, whereas the dimmest cells divided after the antibiotic was removed, suggesting that they were still alive. So there's a difference in single-cell susceptibility to rifampin, and that's predicted by their fluorescence after treatment with calcine. So what's the mechanism to try to figure that out? We took these cells and used flow cytometry to separate out the brightest cells in the population from the dimmest cells of the population, using a population of transposon mutants, so that we could distinguish so that we could look at individual mutations and map them. and we did rounds of enrichment, and we found that the DIM cells, for example, had mutations in proteins like aporin, and aporin is a molecule that allows hydrophilic compounds into the cell from the outside through the hydrophobic cell wall. And that makes sense because calcine has to get in somehow. So porin mutants are relatively dim. And on the other end, we found things like efflux pump mutants, which are involved in getting the calcine out again. And in the absence of those pumps, calcine accumulates intracellularly. And this was we could show that when we isolated individual mutants, actually we constructed individual mutants, we saw that they had different degrees of calcine uptake or accumulation, and we could complement that phenotype. And when we looked at rifampin susceptibility using an assay that is too complicated to explain, it's just not worth it, we found that the rifampin susceptibility was exactly as predicted, which is that those that were the mutants which did not accumulate calcine were relatively resistant as compared to the mutants that did accumulate calcine. So this makes sense. There's a porin involved in calcine and rifampin getting into the cell, and there's a pump involved in getting calcine or rifampin out of the cell. So are there other kinds of mutants you can find? Well, to look for those, Hesper took the transposon pool, and now she did a slightly more complicated screen. She used the flow cytometer to separate the bacteria into different degrees of fluorescence, different bins. She took 8 different bins, and then she sequenced all of the transposon insertion sites in each bin. And that allowed her to reconstruct the distribution of fluorescence with each mutation. So she could figure out the contribution of each gene to the fluorescence. And she found 3 different patterns. She found along with wild-type cells, she saw shifts to the right, shifts to the left, and then shifts in the distribution of calcium accumulation. Here, for example, is a volcano plot of all of the mutants, including many that she made individual mutations in to validate, and showed that they had altered distributions, and the constructed mutants look like that. They look different from wild type, which isn't surprising. And you could take those individuals and sort them in FACS and show that they had different sorts of phenotypes. And for many of them, what you saw is a shift. You saw a shift in the fluorescence either to the right, meaning that they were brighter than normal, or to the left, which means they were dimmer than the wild type cells. But there was one striking mutant in the population. There were a couple things. There were those that had wider distributions. In other words, there was more heterogeneity, and that occurred with porin mutants, for example. And then there was a single mutant that had less heterogeneity. In other words, the distribution of fluorescence was tighter. And that was in a gene called MMPS3. Okay. So what does MMPS3 look like? Well, it turns out that the calcium distribution is different. And I'm going to have to do something incredibly tricky here. Remember, this is wild type. I showed this one to you before. And for the life of me, I can't get the movie of the mutant to work, so I'm going to go switch. OK, stop whatever you're doing. I'm going to go switch. No, I'm going to go switch to a movie which disappeared. OK, it's really little. Let's try it again. OK, what you can probably see, I think, is that the progeny now are much more similar to one another. It went away again, didn't it? OK, that's it. That's all you're getting. The progeny are more similar to each other, and I guess we can, whoa, that's just like some random slide, isn't it? Let me go back to where I was. Okay, I'm not so good at this giving talks thing. I don't know if you've noticed. Okay, so the cells the distribution of fluorescence is much more symmetric at the time of division, so that there's a 60% increase in symmetry. In other words, the cells are more like each other when they divide. And based on that, Hesper renamed the protein the loss of asymmetry mutant A, or LAMA. So, and why did we call it llama instead of MMPS3? The eminent Dr. Barry had named MMPS3 actually as part of the Genome Annotation Project. But MMPS3, which is a membrane protein, is different from all the other MMPSs, which are small membrane proteins. All of the others are paired up with a large membrane protein called MMPL, but MMPS doesn't seem to be. So it's something different, and what's different about it, we find, is it's involved in generating heterogeneity. And what's it doing? I'm not going to show the data for this, but let me tell you that there's something very strange that it does. Mycobacteria are weird. They grow out of one side, so that they extend out of that side and then they put a septum down to create new cells. So at any given time, they're really only growing rapidly from one end. But in the llama mutant, it grows from both ends. And then it lays down a septum somewhere in between. And what that ends up doing is making the cells of daughter cells of similar sizes to each other. So they're much more similar than Mycobacteria, which are highly asymmetric cells. And how does that happen? Well, cell division is driven by these protein complexes that are involved in elongation and division. And unlike E. coli, both of these processes are occurring at the same point in the cell, so they have to somehow separate those. And it turns out that llama, by protein pull down, is associated with members of the elongation complex. And it looks like it might inhibit recruitment of some of the elongation components to one end of the cell. So they prevent, it's a negative regulator of growth at one end of the cell. If you get rid of it, now you get more symmetric growth out of each of the cells. Okay, so that's an interesting piece of cell biology, and I know it's fascinating to everyone in the audience. What's this mean for TB, though? Well, what it means is that these cells are more similarly susceptible to antibiotics. So that if you kill them, if you treat them with antibiotic, they die at a more similar rate to each other. And that's shown here. Now, it's a little complicated because the susceptibilities are a bit different. But you can take mutants like the porin mutant, which doesn't take up rifampin very well, and it's not killed by rifampin very quickly at all. You can take other mutants like the efflux mutant that's killed rather rapidly. But the llama mutant, which is down here in the brown, that's a little more susceptible, not as susceptible as the efflux pump mutant, but it tends to crash much faster with rifampin. And that's true not for all antibiotics, but for a variety of antibiotics, particularly those that affect the cell wall, drugs like vancomycin or teicoplanin, to some extent with ceftriaxone, a beta-lactam drug. And it's true not only in SMEG, but this is your brief look at TB. But it's true in TB as well. So llama mutants in TB have many similar phenotypes, and they die very rapidly when treated with rifampin or with vancomycin. So some of the heterogeneity in the population is being accounted for by the presence of llama. And it's very odd that this should be true because this is a completely dispensable protein. The cells grow perfectly fine without it. And it's almost as if TB has chosen, or Mycobacteria have chosen, to use this protein just to produce heterogeneity in the population, which might be advantageous when they're encountering some sorts of stresses. Now, is this it? Does this explain all of heterogeneity? No, not by any means, because Hesper has made a number of other reporters, single-cell reporters of things like permeability, looked at drug targets or drug activators and their distribution, the redox state of cells, the ATP-ADP ratio, the NAD-NADH ratio, and all of these vary to different extents. So there's a lot going on to making these cells heterogeneous. But this is certainly one of the major determinants of phenotypic variation. And if you could inhibit it, maybe you could increase the rate of kill of antibiotics, even if you didn't change the susceptibility to antibiotics. And perhaps it would make a difference. And we're just doing the animal experiments now to see if that's true in TB. I'm going to change stories now and talk about tracking TB infection. And unfortunately, all the thunder was already stolen by the panel, so I could just stop now. Well, I'll just do it anyway. This is a project that I co-lead together with my colleague, Sarah Fortune, and we have lots of help from Dirk Schnappinger and his lab, Jane Hill, Leonard Lundblad, University of Vermont, and Veronique Dartois at Rutgers. In our labs, the project is led by a really great postdoc who's done almost all the work that I'm going to show you here, except for the work from Dirk's lab, Jeff Wagner, and with lots of help. The most recent addition to the lab, by the way, Ben Browniker, look him up. He's actually, I'm not kidding, he's a tight end on the Chicago Bears, but before camp, he's working in the lab. So it gets back to the question we were being asked before, how do you test a vaccine? And we already talked about a lot of these things. I would have changed my slides if I had my computer with me. You can measure correlates of protection. Unfortunately, and there has been a lot of work on correlates of protection. This is maybe a month old, this search. There are 139 papers on the correlates of immunity for tuberculosis, and there are beautiful reviews that have been written with figures like this, which mean we don't really know anything. So it's been difficult to find a really good predictor of efficacy in humans. We can certainly use animals, and I am proud to say that my lab has been a big contributor to trying to wipe out mouse TB. And I think this is an epidemic that is going to go away in our lifetimes. But how? In particular for vaccines, there's a real question as to whether mice are going to be a reasonable model for TB. Gail had told us before that most humans are protected against TB, but they don't get sick, but every mouse gets sick and dies. So it's really a very different immune response and different histology. So the question is whether, so we're stuck with humans. And the way we do trials in humans is to do trials in humans. We can do gigantic trials like this one, which was testing a viral vector boosting strategy to BCG. It was a huge trial, and it showed this. And for those of you who ever read oncology papers, it looks like all of them, because these two lines are pretty much superimposable. It didn't work. And Gail already told us it costs a ton of money and a very long time to do. So can we do better? And I have a mild suggestion, which is that we deliberately infect people. Okay, let me keep going. Well, the truth is, we deliberately infect people with lots of pathogens. All of the ones listed up here, some of which are kind of dangerous. I mean, cholera you can cure. Gonorrhea is a little unpleasant. It's unbelievable you can get volunteers for those studies, by the way, isn't it? RSV, though, can cause lasting effects. And we certainly infect people with RSV. Maybe the most common challenge is, I love this, there's a place in England called Flu Camp, which you can sign up for to get a chance to be infected with flu and have various things tested. And apparently it's really nice. There are testimonials on the site. You get an Xbox, and you have these friendly people in BL3 garb visiting you as well. So there are pathogens and relatively and modestly dangerous pathogens that are used in people to test vaccines and test therapeutics. So can we challenge people with TB? There are obvious problems with that. It's not that safe. The infection is chronic. It lasts for a lifetime, and it can reactivate decades later. And as we already heard, there's a huge problem with diagnosis. We can't even tell that someone has TB all the time. So that's a problem. Now, we know that there are safe TB-ish kinds of things. BCG is given to infants, and it's tolerated extremely well. There are oxytropes that are probably not in people that are probably safer than BCG, and there are regulatory mutants that I think have gone into people, at least in phase 1 trials, which are not quite as safe as BCG probably, but people are putting attenuated TB into people all the time. The problem is that the most attenuated strains barely grow or don't grow at all. And the very safest ones are oxytropes, which don't replicate in the host. Now, if you're testing a vaccine, that's a problem, as we'll talk about in a moment. It has to grow. So how can you measure vaccine efficacy. You have to get some growth out of a strain. So you'd have to get an infection which grows and then dies. So how can you do that? I'm going to tell you about two different strategies we're trying to do to make a human challenge strain. One is a kill switch that's on a timer. So this switch is off in vitro, so the bugs continue to grow, but it gets turned on during infection. And there's a fail-safe mechanism that we'll talk about in a moment. This is work from Dirk Schnappinger's lab. He's used a regulated system to produce a toxin. There are several toxins he's produced. When I say toxin here, I'm being generic, but he's used phage lysis genes, DNases, all kinds of things that will reproducibly kill the cell once they're expressed. Now, of course, they can't be expressed in vitro, so he uses a tetracycline regulated system. This system is off in the presence of tetracycline, but it turns on when you remove tetracycline, and you get transcription of the gene. And the reason he's gone that way is you could imagine that in a person, you could grow these bugs in the presence of tetracycline. You could even treat the person with tetracycline to keep the bugs around. But as soon as you stop treatment, the bugs die. And this works if you take a plate where you put tetracycline on a disc sitting in the middle of the plate, then the tetracycline-dependent strain will grow near the disc, but nowhere else. Now, we've also taken a second strategy in our lab, which is I'm going to call a slow starvation system. And to do this, we use non-canonical amino acids, which are amino acids not found in nature. This is work really pioneered in Pete Schultz's lab. And that allows us to make a sort of auxotroph out of any gene that we want. So how does it work? Well, if you have a gene where you have a stop codon stuck somewhere in the middle there, the red codon, then as the ribosome goes along, it happily translates, and then it stops when it hits the stop codon. So if you put in a tRNA that will bind to that stop codon and instead insert an amino acid, then you can suppress that stop. What Pete's big contribution is to figure out that these suppressor tRNAs can, instead of carrying a normal amino acid, they can carry a non-canonical amino acid, an amino acid that's not found in nature. And with the appropriate tRNA-tRNA synthetase pair, you can get incorporation of these things. And so what happens in the presence of the tRNA synthetase, and the non-canonical amino acid is you can, at some efficiency, keep going, read through and incorporate that non-canonical amino acid. And that works well. We've shown that with a couple of different genes. This is back to Mycobacterium smegmatis again. We can show that we don't get any growth in the absence of the addition of a non-canonical amino acid, but the bugs are able to grow and grow pretty well when you add the non-canonical amino acid. The same is true. The last gene was alanine racemase, an essential gene involved in cell wall synthesis. Biotin synthetase, which is involved in making biotin and therefore involved in lots of carbon additions in the cell. Once again, a control strain grows, but the mutant strain only grows in the presence of the non-canonical amino acid. So we could get some growth out of these cells. How much growth do we need? We don't know. If normal cells grow like this in the host, and auxotrophs die immediately, we need something in between. We need something that dies sometime after the establishment of some sort of immunity, maybe, or maybe sometime before that. But I think that still we are willing to listen to people in the community to try to figure out how much we need, because it's a lot of engineering. Now, the nice thing about these sorts of strains is that we do get some growth out of them. So if you have an auxotroph, it's a strain that requires an external nutrient, then they'll grow when you add the nutrient. But as soon as you take the nutrient away, they don't have any reservoir, right? And so they drop dead. But this is a little bit different. It's a protein fuse. So in these cases, the bugs are making an enzyme, and we try to overproduce that enzyme, in fact, and it stores it up. And so when the nutrient is gone, or when the non-canonical amino acid is gone, then the bugs will continue to grow until that protein either turns over or it's diluted out to some critical concentration where you can't make the nutrient anymore, and then they drop dead. So you get some growth, and we can show that here, and I'm afraid I'm not good at movies. All these movies are going to show at once, so let me tell you what you're going to see, which is that control cells grow, just continue to grow, and they'll take over the flow cell in a short time. These cells have been grown in the presence of the nutrient. This is alanine racemase, so this is D-alanine. These have been grown in the presence of D-alanine, but there's no non-canonical amino acid. And then at the start of this movie, it's washed out. And these cells have been grown in the presence of the non-canonical amino acid. And then it's washed out at the beginning. So that we're comparing these two, really. So these guys had just D-alanine, and these guys had the non-canonical amino acid. And let's see what happens. Okay, so these guys are growing, and they're going to grow and grow. These guys didn't do anything. These guys have grown some, and then you can see they're labeled with GFP. You can see them flashing off as they die. And if we wait long enough, all these cells will die, as these cells have. But it takes a far longer time for these guys to die than for these. And in fact, they continue to divide for a while. So how long precisely, we don't know. We're playing with that now, and we have different ways that we can modulate that. But it's certainly something we're working on. Okay, I screwed up the slide, so there are no labels on anything. So you can ignore all the labels. And let me just tell you what this says, which is that if you take the timed kill switch strains, one thing that we really worry about is how frequently will they escape, because once they escape, they become fully virulent. And these guys escape at a rate of about 1 in 10 to the 7th, actually a little less, more closer between 1 in 10 to the 6th and 1 in 10 to the 7th. The non-canonical amino acid systems, they escape at a rate of about 10 to the minus 7 to 10 to the minus 8th. But if we combine them both, we don't get any escapes. And we get a theoretical escape rate of somewhere down around 10 to the minus 14. So if we put them together, we think we can make a very safe strain. There's still a lot of engineering to do, and there's a lot of moving this to TB to figure out. But I think that part is doable. But once we have these, how do we tell that the vaccine worked? TB is an infection deep in the lungs. We can't really even diagnose it. How are we going to know whether or not there was a favorable response in a vaccinated individual? And it would be great if we could measure some bacterial component, but we don't yet know of a component that we can measure sensitively to determine what the bacterial load is. So instead of doing that, we thought, how about if we, instead, we're engineering the bacteria anyway, what if we engineer a molecule into the bacteria that we can measure, and that use that to measure the number of bacteria? Now, what would we, anything we use would have to be something that's simple for the bacteria to make, so there's not a huge genetic cost, fitness cost to making them. It has to be safe. It's something that we have to use in humans, so it has to be something we know we can get by the FDA. And it has to be easily detectable, somewhere accessible. Now, one of the nice things about TB is it infects the lung, and what's very accessible in the lung is the breath. So the first thing we thought of is, what if we used volatile molecules? What if we engineered the bacteria to make volatile molecules so that we could detect them in the breath? We tried to make several of them. We have had limited luck with several. We thought we had grape, but it turned out to be a contaminant. But so the one, and we're continuing to work on these, but the one that we've had the most success with is methyl salicylate, which gives wintergreen its flavor and smell. Methyl salicylate is made by a fairly simple biosynthetic process from charismate. There are only 3 genes involved in its synthesis, and it smells nice. And when you engineer bacteria, you can make Mycobacterium smegmentis, which actually the flasks are minty smelling. It's sort of minty smelling old sneaker smell. And It makes a lot, so much so that if this is work from Jane Hill's lab, this is Ted Miller's work, it's so much that when you do GCMS on the headspace of these cultures, the very largest peak by a large margin is this one, which is methyl salicylate. All these other things are endogenous to the bug. So it's not present in the empty vector, but present in the wintergreen cells. And we can measure this relatively sensitively. We think we can do even better than this, but right now we're detecting down to a limit of about 10 to the 4th bacteria. We think we can do somewhat better than that, though maybe not a lot. It's great to put it into SMEG, but we can also make Mycobacterium tuberculate. And it too makes a very large amount of methyl salicylate. So we could easily smell it if we chose to, which we haven't yet. Okay, so what do we do with these things? Now, we'd love to make a human challenge strain, but I think, I think it could be a very useful tool. In the meantime, though, there are other things that this is useful for. In particular, I think this is very useful for animal experiments, and especially expensive animals like monkeys, because non-human primates, if you want to find out how many bacteria there are, you have to sack the animal. But using this system, we may be able to measure the total bacterial burden continuously in the same animal. And the same would be true for even for mice. So we would certainly decrease the number of animals that we'd need. We have a lot more work to do on this, but we are excited about it and also happy to take any suggestions that people have. So the first project I told you about was paid for by the NIAID and Burroughs Wellcome Fund. We have had generous funding from ARIS and Bill and Melinda Gates Foundation, the only people crazy enough to fund the second project. But I will say that they're more than our funders. They are our partners, our intellectual partners in this project. So you can blame them as well as us. Thank you very much. Bhavesh.
So Eric, going to the first part of your presentation, the llama mutant is really cool. So have you looked at sort of what division and elongation complexes are doing? So how does div4a, FTSZ, maybe even some of the peptidoglycan biosynthesis enzymes, how do they behave in terms of localization?
We've only started to look at it. This is a relatively recent finding. That llama is bound to at least some of the components, at least PON-A1. So we haven't really tried to visualize each one of the components at the same time. As you know, a lot of these components are hard to visualize because they're not highly expressed and they're secreted, and the fluors aren't great for secretion. And llama is a very non-abundant protein, so it's very difficult to see, even though it's a multi-pass membrane protein, and you can label it on the cytoplasmic side, but there just aren't many copies of it. So we're working on it, but we don't have great resolution yet.
And then just so in terms of drug susceptibility, you know, to follow up on Sarah's hypothesis that the different size progeny have different outcomes when you challenge them with antibiotics. Have you looked at this at a single cell level, trying to sort the daughters of the llama mutant?
So we see the same in wild type cells. We see the same thing that Sarah sees, which is that one of the progeny of cell division is more susceptible to rifampin than the other. Llama kind of makes it difficult to tell the difference between the two progeny because the cells are now growing from each end and they come out pretty much the same size. So essentially we are equalizing that. I think we're equalizing the susceptibility at the time of cell division with this mutant. Joanne.
All right. So you indicated the llama mutant grows perfectly normally. Grows normally.
Yeah.
Under the conditions you tested.
In vitro, in 7H9 medium, it's completely indistinguishable.
So I'm sure you know what my question is, which is is there a biological role, though, for heterogeneity, i.e. if you stress it or do a competition, or you've alluded to putting it into an animal indoor, what might your prediction be?
So super good question. I think We've tried a number of, in SMEG, not in TB, we haven't done the TB experiments. In SMEG, we've tried a number of in vitro stress conditions, like peroxide stress or hypoxia. These are not such great stresses for SMEG, but no difference. We strongly suspect that there are differences, and I think what's telling is we have done initial mouse experiments, infection experiments, and the bacteria also grow normally in mice, except that they take a hit of 1 to 2 logs upon infection. So when you infect, you see this, you see very few cells establishing infection. The ones that do establish grow normally. I don't think we figured that out yet, so we're trying to.
So in the mutant, the llama mutant, is the growth rate from the pole half?
Yes, it is. So the total growth rate is still the same. And in fact, I'd be interested in what Bhavesh sees. Pretty much all of our cell wall mutants, our cell division mutants, which no longer divide, the total growth rate, even in those cells that are branching and forming those weird structures, the total growth rate is exactly the same as it would be in wild type. So if you add up all the constituents, it's as if there's a limiting factor in how much stuff you can make,
right.
But where you put it is not limiting.
Yeah.
Tom.
I'd just be very curious to see your vision of how this auxotroph could unfold into a true human trial. So would it be...
Oh, we already released it here.
Aerosolized infection, what dose? How would you instill it? What kind of time points?
Yeah, I think the answer is yes. So what we're thinking now, and we don't know the performance characteristics of what we're going to end up with, what we're thinking now is it would be an aerosol infection, but it would not be a super low dose infection because we're never going to get a sensitivity of measuring a very small number of bacteria. And we want to see some dynamic range. We would like to start at a number of bacteria that we can detect so that we can see the increase above that level. That's obviously problematic in some ways, and this may not be useful because will a vaccine protect you against 10,000 bacteria? That would be quite a vaccine. We're asking a lot for it, of it. It may be that we just have to go let the bugs survive for longer so that we can reach a threshold where they are detectable and go with a lower dose. But I think it's going to depend on how many we can detect when we actually put it into at least an animal, and see what those thresholds are. Part of the reason that we're continuing to look at other reporter molecules beyond wintergreen is we'd like to find something even better. 10 to the 4th is good. We think we can do better with some of the other reporters. The problem is bacterial metabolism. The bacteria we can get them to make some of these things, but they eat them. So they metabolize everything. So we have to do a little bit of metabolic engineering on the host. Okay, thank you.